Good evening. Good evening and welcome to the 2014 Sverdrup Visiting Lecture Series. This series is sponsored by an endowment from the Sverdrup family and the Minnesota NASA Space Grant Consortium. The two goals of the Sverdrup Visiting Scientist Program are, first, to provide an opportunity for Augsburg students and faculty to interact on a personal basis with scientists of national stature. Second, the program provides an annual opportunity where Augsburg and the wider scientific community can expand their knowledge on a scientific topic of national significance. Tonight we are honored to have Squibb Professor in Molecular Biology at Princeton University, Bonnie Bassler. Professor Bassler has a CV too long to recount and distinguished, so we won't. We will only say that we look forward to her insights into the world of bacteria and their tiny conspiracies. Please welcome Dr. Bonnie Bassler.
Oh, well, I guess it's good. So thanks for that accurate introduction. And I'm just delighted to be here. It's a big honor to get to give this seminar, and plus it's a treat. And I just want to say that I was really lucky to get to go to the party beforehand and get a glimpse into the amazing collegiality that exists at this place, the genuineness, the commitment to education and to getting kids in the lab. That was super just lucky that this got to coincide with my seminar. So I feel like I got a good touch of what Augsburg is about, and now I would like to give you a touch of what I'm about, and actually what the students in my lab at Princeton have been doing. And so I only have one goal for this seminar, and that's to try to convince you that bacteria can talk to each other. And if I manage to do that, a sort of secondary goal is to try to convince you that they're multilingual. And then the third thing that maybe will happen by the end of this talk is that I'll convince you that learning that bacteria can talk, learning about these languages could be useful, because maybe we can apply that to make people more healthy or to make products for industry that the world needs. And so that's where we're going in a nutshell. And so so I know that this is mostly about physics, so I want to first remind you I'm a biologist, and first just remind you what bacteria are, just in case it's been a while since you thought about them. So on this picture I have sort of a cartoon of a run-of-the-mill bacterium. This would be like an E. coli that lives in your gut. And all bacteria pretty much look like this. They're single cells, and they're covered with a membrane that you can think of like their skin, and that keeps the outside out and the inside in, the same as your skin does. On the inside, they have the cytoplasm, which is the goop that fills the cells, and all the biomolecules are roiling around inside of this cytoplasm. And so those are all the things that an organism needs to make more of itself and to be alive. But what's important for tonight is this blue stuff. That's the DNA, the genetic information that gives the bacterium both its physical features and also all of its traits, its personality. And what's important about bacteria is that they only have one piece of DNA. So they have one chromosome, and they have a few thousand genes. So they don't have very much genetic information to use. And they're also, I can't do this, I can't do it. And then they're also, so they're pretty stripped down. They're primitive both looking and in terms of their genetic content. They are, however, the oldest organisms on Earth. So we know that they got here right after oxygen got here, so they've been on this Earth for about 4 billion years and have colonized every niche on Earth that we know about. And so they've always, though, because they're so old and so primitive looking, been thought to be sort of asocial recluses. And what I mean by that is if you watch what a bacterium does under a microscope, what they do is that they consume nutrients from their environment, they grow to twice their size, they double all of the products inside of them, they cut themselves down the middle, and 1 becomes 2, and 2 becomes 4, and so on and so forth. And they were always thought, once they divide, to have no knowledge, if you will, of their sister cells. So they simply act as individuals. And I want to try to convince you tonight that that cannot be true. But before I get to that, let me first tell you about your interactions with bacteria, just in case you haven't thought about that very much lately. Okay, so what I've done, this is my view of the world. So what I've done is to make this picture of my idea of a man, and I've used a metric. Each of these circles represents some number of the cells, the human cells, that it takes to make up a human being. And so we know what that is. It's a few trillion cells. So that's how many cells you need to give you your physical form and to make you be alive. At any moment in your life, starting a few months after you're born, you have 10 times more bacterial cells than human cells in you or on you. So if you want to use this metric, at best, you're 90% bacteria, 10% human. But of course, this is the 21st century. It's not about the cells anymore. It's about the genetic information, right? So I've remade my man out of the A, T's, G's, and C's of the famous double helix of the DNA, right? And the human genome has been sequenced. We know how many genes there are. There's about 25,000 genes that give you humanness. So that's how many bits of information you need to be a human. So it turns out you have 100 times more bacterial genes in you or on you every moment of your life, right? So you can pick which metric you'd like. At best, you're 10% human. In my view, you're really only about 1% human. All of the rest of that information comes from these bacteria that are in and on you. And these are not passive riders. It turns out there would be no life on Earth without these bacteria, because these genes that these bacteria provide you provide genes that your genome doesn't have, and you would not be alive without those genes. So now what we know is that there would be no plant, no animal, no insect, no human, no nothing without this consortia of bacteria acting as your 24/7 partners to keep you alive. So that's the way I think about the world. But of course, you've heard about bacteria, and probably what you think about the most is this top part, right? I don't want to stand up here and be a Pollyanna. There are all these bacteria. When you hear about bacteria, it's because they've done something terrible, typically, right? So there's all these bacteria that live in the environment that if they get in or on you, they can cause you to be sick or they can kill you. Right? But now what I hope you'll also appreciate is that what we're starting to learn, and this has only happened in this century, is that all these bacteria that live on us daily when we're not sick, they do these jobs that we need. And so some that you might have heard about is that they digest your food. If you eat salad, if you eat plant food, you don't have any of the enzymes to get the nutrients. Bacteria provide that. They educate your immune system. They keep bad microbes out. They provide you many of your vitamins. And so this list goes on and on and on. And so the problem with bacteria is that you can't see them, right? So you don't realize these bacteria are there. It's not like you get up every morning and thank your bacteria for being alive. When you think about bacteria, it's typically when you don't feel well, and so you think about them historically as pathogens. And so now our view in the microbiology field is changing, and we're starting to understand that, in fact, most of the time that we encounter bacteria, something good happens. And so that's sort of the two faces of bacteria. But what I want to now move on to is the question that has been at the heart of my lab's research for as long as we've been in this game. And that's whether you want to think about all these good things, or you want to think, all these good things bacteria do, or you want to think about all the bad things these bacteria do. The question that we have is, how can they do anything at all? They're puny. How can it be that these bacteria can either kill you or conversely keep you alive? And so what we want to understand is, how do bacteria get any bang for their buck? And what I want to try to convince you is that the way that they do these miraculous, wonderful, and terrible things is by talking to each other, by counting each other, by recognizing when they have the right number. If they all do something together, they can accomplish tasks that they could never do if they were individuals. And so that's where we're going. And so what I thought I would do, since it's a special seminar, is give you a sort of historical tour of how we got to this idea. And it all started with this animal, which is the Hawaiian bobtail squid. And so this is an animal that lives in the ocean. It's about this big, full-grown, and it lives in a beautiful one-to-one symbiosis with a marine bacterium named Vibrio fischeri. And Vibrio fischeri has the special property that it's bioluminescent. And so the squid needs the bacteria, and the bacteria needs the squid. And so first I'm going to tell you about the squid. Okay, so this squid is nocturnal, and so what we've done is to put it in an aquarium with the lights on, but there's no sediment, and so it looks kind of spastic and nervous, and that's because during the day, this squid sleeps buried in the sand to escape from predators. And so when we put it there, it looks kind of upset. And so now, if we give it sediments, this is what happens, is that the squid hunts at night for food, but then every morning it buries itself in the sand, as I just told you, to escape from things that would see it and eat it. It's thorough. You only get eaten by a predator once. Okay, so he's almost done, and so now he's invisible. So that's a fantastic strategy to escape from predators during the day, right? So if I showed you this aquarium, you wouldn't know that there's a squid in there. There's actually about 10 of them in there. Okay, so the strategy from the squid's point of view to stay alive is to bury yourself in the sand in the morning, every morning when the sun comes up, and escape from predators. But the problem is that it has to eat, and so that's this nighttime business. It needs to come out at night in order to hunt for food. And so the squid lives in just shallow water, so just a couple feet deep of water. And so the problem for the squid is that because it's in such shallow water, on bright nights when there's lots of starlight or moonlight, that light can penetrate the depth of the water that the squid lives in. So if it was swimming around, it would make a shadow, and predators could see its shadow, calculate its trajectory, and eat it. And so this is where the bacterium Vibrio fischeri comes in. So what the squid has evolved is a specialized light organ that houses this bacterium Vibrio fischeri. And now I'm going to show you that. We've turned the squid on its back, and I hope what you can see are these two glowing orbs or lobes of this specialized light organ. And so the way that it works is that this light organ is under the mantle or the body of the squid. And inside that light organ is Vibrio fischeri at something like 10 to the 12 cells per milliliter. It's like toothpaste of bacteria. And they're in there, and I told you they have this special property that they make bioluminescence. So they make this blue light. And so what happens is when the squid emerges at night to hunt, it has detectors on its back, so it can sense how much starlight and moonlight hits its back. And then it opens and closes a shutter. It's just the squid's ink sac over this light organ. So it detects how much light hits its back, and then it lets out, made from the bacteria, the same amount of light underneath it. So when it swims, it counter-illuminates itself with the light from the bacteria so that it stays invisible from these predators. So this is sort of like the stealth bomber of the ocean, right? It cloaks itself in this invisible device, and it's got this strategy for at night, and then I showed you the strategy for the day. Okay, so that's why the squid is willing to feed these bacteria. So what's in it for the squid is protection. What's in it for the bacteria is food. The bacteria cannot grow to 10 to the 12th cells per milliliter in the ocean because that's nutrient poor. So in this light organ, it's filled with all kinds of goodies that the bacteria can eat, and they can make more of themselves. So that's the evolutionary selection from the bacterium's point of view. And so now for the rest of the talk, I want to talk about bacteria. So what about the bacteria? It turns out that they only make light at night. And so how do they know that? How do they know they're in that light organ and when it's night? Okay, so that's because they talk to each other. So first, how they understand that they should make light. So it turns out that the bacteria only make light when they're at high cell number, and that only occurs in the squid and only at night, and I'll tell you how we get there. So first, how does the bacteria know whether it's alone or whether it's at high cell number? It's because it talks with the chemical language. So here's my cartoon of this Vibrio fischeri cell, right? It's tiny, right? The world is big. And so if it's alone, when it's dilute, it wants to have the program of gene expression going that's good for acting as individuals. So it has some subset of its genes on, and it's doing some behaviors. And among the things that it does is it makes and releases a small molecule that we call an autoinducer, and you can think of this like a hormone. And so what happens is when the bacteria are alone and they make and release this molecule, it just floats away, and so the bacteria can't detect it. And so since they can't detect that hormone, that tells them you're an individual. In this case, don't make light. But then as the bacteria grow and double, as I told you that they do, since all the bacteria participate in making a share of this hormone, this autoinducer, the extracellular concentration of that autoinducer increases in proportion to cell number, right? So the more cells there are, the more of that stuff, the molecule there is. And it's when this molecule hits a particular threshold level, the bacteria detect it, and they use it as a proxy to know that there's neighbors around, right? So they detect this molecule, and when they do that, in response to it, simultaneously, all of the bacteria turn on the genes that make the enzymes that make light, and they do it in unison. So the way the bacteria know when they're alone or when they're together is by virtue of whether or not this molecule is there. And so that only happens in the light organ of the squid, because that's where the bacteria can grow to this high cell number. The molecule is trapped there with the bacteria. It builds up. It tells the bacteria, you're not in the ocean, you're in the squid, you should make light. Okay, so everybody gets that part. And then the reason it only happens at night is because, now the squid controls this part, is every morning when the squid buries itself in the sand, like I showed you, it's got a pump that's attached to its circadian rhythm. So when the sun comes up, the squid pumps out like 95% of the bacteria. So just a few get left in that light organ, but they're dilute. And so they turn off light. As the day goes by, they grow and they double. This molecule builds up, and so the light comes on exactly when the squid needs it every night, and nobody wastes any energy. Okay, so that's the selection from both points of view, right? But what was wonderful for us about this bioluminescence is we could just see it. If we grew the cells in a flask, we could just see it. And what we could see is that when the bacteria were dilute, they didn't make light, and when they grew to a certain cell number, they all turned on light together. So this bioluminescence made this invisible world of bacteria visible to us. And for the first time, this is like 25 years ago, we thought maybe bacteria have a way to do collective behaviors. The other thing we could do is we could spin the cells out and take the cell-free supernatants from cells like this and just squirt them on to these cells, and the bacteria would turn on light. So it was clear they didn't know how many cells were around. They were measuring something that was in the liquids. Right? So that was our way in to understanding the beginnings of this idea of bacteria being social. So then we brought the tools of molecular biology and genetics to it to find out what are the genes and the proteins involved in this system. And so now, again, this is supposed to be a cartoon of a Vibrio fischeri bacterial cell. What we found was there was a gene and an enzyme that made that hormone, the autoinducer. And that simply freely diffuses. It just leaks out of the cells. So the more cells there are, the more of this hormone there is. What we also found was that there was a receptor protein that sits on the bacterial cell surface. So it has a part of the protein on the outside and a part of the protein on the inside of the cell. This is exactly the way your hormone receptors sit on your cell surfaces. They sit there and they wait to bump into a molecule, in this case the autoinducer, and when the autoinducer slots into the outside portion of the protein, the inside portion of the protein sends a signal down to the DNA to turn on the genes that make the enzymes that make light. So this is really simple, right? The more cells there are, the more of this autoinducer there is. At a critical amount of that autoinducer, it can find that receptor protein, send information into the cells, and in unison, all of the bacteria turn on light. And so why is that interesting if you care about things other than bioluminescence? This was the clue that bacteria might do this, right? But then, of course, we started to think, well, why should this be restricted to just this goofy bioluminescent bacteria? And so now, in the past 20 or so years, hundreds and hundreds and hundreds of these communication circuits have been found in all kinds of bacteria. So now we have probably 1,000 cases of bacteria that have an enzyme that makes a hormone molecule. There's always a partner receptor, and then together these complexes turn on all kinds of genes that bacteria want to express when they're in a community, but not when they're alone. And so now we have a fancy name for this process. We call it quorum sensing. The bacteria vote with these chemical votes, the vote gets counted, and then everybody goes along with this democratic decision-making process, right? And typically, and so it's not just about light. This is all over the bacterial world. This is the norm. And typically, in every species that we look at, something between 200 and 600 genes are controlled by quorum sensing. So the idea is when a bacterium goes from acting as an individual to acting as part of a collective, it's a big developmental process that takes hundreds of genes to get there. Right? So that's how it works. We also know a lot now about the molecules that are involved, right? So on my triangles, on my slides, I've been showing them as triangles, but this is the actual molecule, the word that Vibrio fischeri talks with. So what you can see is there's a ring, and then these are just carbons. And so, of course, we've known what that molecule is the longest. That's for historic reasons. But then lots of other scientists started purifying these molecules from other species of bacteria. And so what I've done here is to simply put a smattering of different quorum-sensing bacteria and the molecule that each one of these bacteria uses to communicate. And what I hope you can see is that all of the molecules are related. They all have this ring on the left side, but then these carbons are slightly different on the right-hand side. And what that does, those tiny differences, is to confer exquisite species specificities to these molecules. So see how they look sort of like keys To me, they are just like that. Each of these slots into their partner receptor and sends information in, but there's no crosstalk, just like the keys that you use for your locks. And so what I mean by that is if we take the molecule from Agrobacterium and put it on Vibrio fischeri, nothing happens. And likewise, Vibrio fischeri, or Agrobacterium is impervious to Vibrio fischeri's molecule. So what we understand now is that these molecules allow intra-species communication. These are private, secret conversations that bacteria have with their brothers and sisters so that they can decide when there's enough of their kin around that they should all do something and they get this collective success. So that's quorum sensing. We also now, besides knowing how these circuits work, we also know a lot about these behaviors. And so I told you in detail how this works for Vibrio fischeri and why it's only useful to turn on light as a group, right? So that's how the field started. But then it moved actually very soon after that into the idea of what about pathogenesis? So I've already alluded to this idea. How can it be that these little bacteria make you sick? Well, the reason is because they make you sick as a group. So I'll just give you a couple examples. So Pseudomonas aeruginosa is an opportunistic pathogen. It just lives in the dirt. You and I encounter it every single day of our lives. It doesn't hurt you unless you have cystic fibrosis or you're immune compromised. So you probably know that people who have the disease CF have a genetic mutation in their lungs, so they can't clear their lungs. You and I breathe in all kinds of stuff, and we have mechanisms to keep our lungs sterile. People who have CF are constantly colonized with bacteria. And what happens, for reasons that we don't understand, is typically when a person is in his or her teens, they'll become permanently colonized by Pseudomonas aeruginosa. They'll have that infection for 20 or 30 years, and they will actually die from the Pseudomonas infection. So people who have CF die from Pseudomonas infection. And the reason is because Pseudomonas has quorum sensing. So what happens is when the bacterium gets in, it sits down, it adheres to the lung cells. That's called what we call a biofilm. So it makes a film on the lung cells. It secretes this polysaccharide matrix that encases the bacteria and makes it impervious to antibiotics. And so the bacteria grow inside this film, and then simultaneously they all release 20 or 30 or 40 toxins or poisons that actually are the things that make the person sick. They're because they eat up the lung tissues. The bacteria are just trying to get food, just trying to get nutrients. But the consequence is that the person gets this terrible infection. And so I think that you can see from this Pseudomonas example is that it is not useful for a single bacterium invading a host to release, to dribble out a few molecules of toxins. First of all, they would have no effect because the host is huge. And second of all, if I release my toxin, I will never get my own product back. Only when the collective releases these products, these exoproducts together, do I get the benefit of your work and you get the benefit of mine. And so now we have dozens and dozens of cases of clinically relevant bacteria that if you make a mutant so that they can't talk or they can't hear, they are avirulent, even though they have their entire repertoire of toxins intact. They simply don't know when to release. And so now we get that quorum sensing is a central component of these two steps in an infection: making the biofilm, so sitting on the surface, that takes the community to do, to make this sort of community, and then to release their virulence factors in unison. I put this other one, Erwinia, on this because this is a bacterium that you have heard of. So you know how if you leave your lettuce in the drawer of your refrigerator too long and it turns into that soup? That's Erwinia. So this is a pest in agriculture, right? It does the same thing as Pseudomonas except to plants. It makes a biofilm on the plant, and then simultaneously the bacteria release these toxins that make a wound in the plant. What else it does that's really insidious is simultaneously to releasing all of these virulence factors, it actually releases a whole raft of antibiotics. And so what it does is it has immunity to those antibiotics. So it kills off all the competitors to keep the wound for its brothers and sisters. And so I hope you can see from these examples that any time a bacterium is going to give something away to the world, it doesn't have a chance of getting it back. So now what we get is that quorum sensing controls all of these kinds of behaviors, that it takes lots of bacteria working as a collective, and only then does the behavior become successful. We're now starting to learn that all these beneficial behaviors that I told you about are also controlled by quorum sensing. It's just that we haven't known about them as long, and so historically the field has focused on pathogenesis. But it's becoming very clear that these good things that we like bacteria to do, which keep us alive, are also controlled by quorum sensing. So once we sort of got that far, we started to get this idea that bacteria act in groups, we took a step back and we said, you know, it's really great if you're Vibrio fischeri and you live in this pristine environment of this squid and there's no other bacteria there. But that's not actually how most bacteria live. I already told you most bacteria live in a biofilm, and this is a picture under a microscope of a biofilm. This is human skin. You are covered with a biofilm all the time, and then what you can see is every one of these different shapes and sizes is a different species of bacteria. And so what we started to wonder, if quorum sensing is really about counting your neighbors, and these languages are so species specific, how do bacteria take a census of the community when there's all these other species around? And so we wondered if quorum sensing couldn't be more sophisticated than we were thinking about in the beginnings of our studies. And so what we decided to do to explore that question was to work on a cousin of Vibrio fischeri. And so this is a bacterium that's named Vibrio harveyi. It's a bioluminescent marine bacterium, but it lives free living, and it makes light in the free living state. And so we thought, well, how does that one control quorum sensing, given that it encounters hundreds or thousands of other species of bacteria, unlike Vibrio fischeri that's living in the light organ? And so the reason that I put this slide up here, besides the obvious lab propaganda, to show the students what graduate students do with their time, right, was to just tell you how we do these experiments. So I'm a geneticist, right? And so what you're looking at, right, we want to know how is quorum sensing controlled, right? And so what was so fantastic about these bioluminescent critters, right, is you could, as I told you, you could just see them. And so this picture, to take this picture, all we did was turn the lights off in the room, and we simply took the picture from the light the bacteria are making. So we haven't done anything to them. This is just what Vibrio harveyi looks like in a flask and on Petri plates. And so then what we could do, and this is what a geneticist does, is we could simply make random mutations in the Vibrio harveyi chromosome, turn the lights off in the room, and look for ones that were dark. And by doing that, that's how we got the Vibrio fischeri genes and how we're going to get the Vibrio harveyi genes, right? So it's really simple. You just look for guys that are goofy for bioluminescence because we know they have to talk to each other to make that light. Okay, so everybody gets that experiment, right? Okay, so we did that, and what did we find? Well, certainly, of course, we found the quorum sensing system, so we found that there was the enzyme that made the signal molecule. I actually showed it to you a couple slides ago, and then there was this partner receptor. But what was interesting was when we knocked out these components, this bacterium still had quorum sensing. And so we went hunting some more, and what we found was that there was a second enzyme that made a second autoinducer molecule, and it had its own partner receptor. And then together, all of that chemical information came into the cells to tell the cells to turn on light, and hundreds of other genes, but we're simply measuring light production. So there were two systems running in parallel. And what we wondered when we found those components is, well, what's the point of having two systems? If these two molecules don't encode different pieces of information, having two quorum sensing circuits is not better than having one. And so we wondered, what's the point of this second circuit? And so we had cloned the genes for this, and just like we have the human genome, we have thousands of genomes of bacteria. And so what you can do is you can take your favorite gene, plug it into the database, and ask, does anyone else have it? So we had the gene for this enzyme. We called it LuxS. Everything's called Lux for Luxor, the god of light. LuxS for the synthesis of this molecule. So we took this LuxS gene, we put it in the database, and lo and behold, what we found was that all other bacteria have that gene as well. And so this is just a list of some of the bacteria that have a highly conserved LuxS gene. Right? And so I don't know who you want on here, but of course it's a who's who of pathogens, right? So this is, that's bad breath, that's anthrax, this is bubonic plague, cholera, staph, strep, right? It was all of these pathogens. And so then we showed that every one of these bacteria and others makes this second molecule as well. And so then we asked, well, what are these bacteria doing with it? They're not turning on bioluminescence. Bubonic plague doesn't have bioluminescence, right? And so what are they doing with this second molecule that I'll call autoinducer 2? And so we and lots of other scientists started to ask, what are the traits that are controlled by this new molecule? Oops, what happened? Oops. And so here's just, again, a list of different bacteria that different scientists have studied. And what you can see is that in every single case, what's controlled by this new molecule is biofilms, how bacteria sit on these surfaces in groups, and then the group-wide production and secretion of virulence factors. So just like this autoinducer 1, the second molecule seems to be playing a role in virulence as well. And so then we wondered, well, what is it? And so we purified autoinducer 2, and this is the molecule. It's a 5-carbon molecule, and what's important for this part of the talk is not what the molecule is, but that there's only one molecule. So no matter who we purified this from, every bacterium that has LuxS that makes this molecule makes exactly the same molecule. So unlike in the first system where each molecule was slightly different, in this case, all the bacteria make the same molecule. So what we think then is that autoinducer 2 is a non-species specific molecule. It's sort of a universal molecule, like the trade language or the bacterial Esperanto that allows bacteria to talk across species. So I can talk to you and you can talk to me because we use the same word. And so now what we're convinced of is that bacteria have to at least be bilingual. And I will not be out of a job when I go home tomorrow. There are more molecules to find, but at least they have to have 2 molecules. So they have one molecule that allows them to count their siblings. And then they use autoinducer 2 to take a census of everybody else in the environment. And so the computation that we know bacteria do now is the following. What we think they do is the first thing they do is they simply ask, am I alone or am I in a group? So they have their receptors out there and they're just looking around, if you will, for molecules. So first they do the simple question, am I alone or am I in a group? And so they scan the environment for any of these autoinducer molecules. And that begins to set the program of behaviors that they're going to carry out. Individual behaviors or group behaviors. But then beyond that, what we've shown that they do is that they actually measure the ratio of these 2 molecules. And then they tailor the program of genes that they turn on based on who's winning and who's losing in any given consortium. So if they see a lot of their siblings around, they carry out a certain set of behaviors. If they see a lot of the enemy or their competitors around, in fact, they change which genes turn on and off. Right? So what we believe then is that bacteria have the capacity to distinguish self from other, and then they can do something about it. A lot like what we think higher organisms can do as well. So that's how these circuits are set up, and what I thought I would do to finish is then promise you 3 things. They talk to each other, they're bilingual, and then, well, okay, so now we've learned bacteria talk with these chemicals. We've learned that this is essential, this communication for virulence and for beneficial behaviors. So can you do something about it? Right? So now there's lots of people in the field wondering, could we manipulate these quorum sensing circuits to make either new kinds of antibiotics or new kinds of products for industry? And so I'm sure you guys know we have this global problem that there are no antibiotics left in our arsenal. The bacteria have become multi-antibiotic resistant. And so the question in my field is there another way to think about antibiotics? So antibiotics kill bacteria. They either slow their growth or they kill them, right? So then when bacteria get resistant, they have a big growth advantage because everybody else is being killed. And so the question is whether you can do behavior modification. Can you actually just get them to either be quiet or be deaf so that they don't understand to release their virulence factors and buy the immune system time to clear them, which is what your immune system is doing with bacteria that don't have major virulence factors. So we want to try that. And so there's sort of 2 strategies that have to do with the 2 different molecules. So the first one is to try to interfere with this species-specific system, right? You have a Pseudomonas infection, you have CF, we want a Pseudomonas-specific drug. So can we take advantage of the little differences between those molecules I showed you to make what we would call a narrow-spectrum therapeutic? So that's one possibility. The other possibility is to just go against, try to interfere with this broad-spectrum system. So sometimes you go to the hospital or you go to the doctor and you don't know what's wrong with you. You don't know what pathogen you have. And so what you get treated with are broad-spectrum antibiotics while your physician tries to narrow down what you have, and then you get narrower and narrower spectrum antibiotics as the physician understands the disease. So maybe we'd like to go to try to interfere with this system that's generic in order to make broad spectrum therapeutics, right? So we're trying both of those strategies, and the logic for how one does that is identical. So I'm just going to show you one story. And so tonight I just picked, because we have a brand new molecule, I picked how we try to find a molecule that interferes with this intra-species communication system. Okay, so in my cartoons, I use a triangle, but I've already showed you the molecules, right? So this is actually the molecule that Pseudomonas, or one of the molecules Pseudomonas aeruginosa, communicates with. And so we would like to make a molecule that looks kind of like this, but that interferes with reception of this molecule. So using chemistry, by my cartoons, what we'd like to do is to take this molecule and put some bumps on it, right? So we can add a couple of carbons, we can make a ring, add a sulfur, add a nitrogen, right? So we have a key, and we want to put some bumps in the key, right? So that when it locks into the receptor, it just gets jammed, right? So everybody understands that, right? So we're trying to make an antagonist, right? And so that is what one can do, is we can make molecules using synthetic chemistry. And then also what's been wonderful in the last 5 or 6 years is that the National Institutes of Health has collected up all kinds of molecules that chemists have made for whatever reason. So they have a library of hundreds of thousands of molecules that all have a different shape. So what one can do as a scientist is to just screen through that library for any molecule that fortuitously has the right bump on it and is an antagonist of our signal. So we can either make them one by one, or we can just get our hands on these whole libraries. Okay, so we have, if you believe me, access to the molecules. The question is, how do you get the ones that interfere? I mean, most of these molecules won't do anything to quorum sensing. And so what we decided to do is to go back to these bioluminescent bacteria, right? So you'll remember that they require those molecules to turn on light. And so what we can do with all these synthetic molecules, or the molecules in the library, is one by one, add those new molecules and ask, does Vibrio harveyi go dark? Right, so everybody gets that, and you do this with robotics. You can look through hundreds of thousands of molecules in a week. And so what we can do is to go from a few 100,000 to down to about a dozen that look useful in a week. And then we pick the best one, and I will show that to you. So here's the molecule that was the best inhibitor that we got out of the screen, and it should be obvious to you why it's an inhibitor, right? So it's the signal molecule with the big bump on the end. So it's like a key with a big bump. And when this molecule slots into that receptor, it blocks access of the real molecule, but it doesn't send the information into the cell. Right? So now we have an inhibitor, and it shuts down light in Vibrio harveyi. It turns out no one actually cares if I can shut down light in Vibrio harveyi. That's sort of something I've learned over the last 20 years. What they really care about is this, right? So we got the molecule from harveyi, right? And then we had to put it into a mammal. And so we have an animal model for cystic fibrosis, right? And so if you take the pathogenic bacterium and you give it to the animal dies because it has cystic fibrosis. And so the question is, well, what happens when you give the infection, but you give it with that antagonist that I just showed you? And it turns out that, in fact, the animal lives. And so we're actually getting there. These are not medicines yet, right? That molecule that I showed you is straight out of a library screen. What medicinal chemists have to do is to build in potency. It needs to be much more potent. They also have to build in safety, right? Like right now you'd have to take a pill this big and it would kill you, right? And so in fact we have to do a lot of medicinal chemistry. This is what you would call a lead molecule. But what it did for us is to give us confidence that there's merit to the idea of trying to interfere with this behavioral process to make new kinds of therapeutics. And so now that we have the molecule I showed you, we are very busy working with medicinal chemists to try to make that molecule or some other molecule, right, into something that looks medicine-like. This one doesn't. But it was a good proof of concept for us. And so that's where we are right now in these quorum sensing studies. And so I hope what you think after this talk is that, besides my talking, is that bacteria talk to each other. They don't have voices, and so what we think that they use is chemicals. There may be other mechanisms for them to communicate, but for sure there is a rich chemical lexicon out there, and there are many more molecules to be discovered. This is a very young field. I hope what you also think is that bacteria are multicellular, right? They carry out these collective behaviors. So what I would like to argue is that this is 4 billion years old. This idea of being able to work in groups did not originate with higher organisms. Bacteria invented this. And what's wonderful about studying the actual biochemistry of how these circuits work is that the principles that are embedded in these circuits go up through evolution. And so by studying these sort of simplified collective behaviors, we hope we'll be able to help our colleagues who study collective behaviors like how your organs work, right? And give them insight into their studies as well. I hope what you believe is that bacteria can tell self from other by using these two molecules, one that says me, one that says not me, right? And again, we would argue that's what happens in the human body. It's not like your kidney cells get all mixed up with your heart cells every day, and that's because there's different hormones, there's different chemicals that tell cells inside a bigger organism what jobs to do. Again, we think those principles are embedded in these ancient organisms, and that by studying how these bacteria distinguish between these molecules and how they do what they do in response to these molecules, we'll be able to give insight into higher organisms, and especially when something goes wrong, right? To maybe have something to say about diseases of higher organisms. And then, of course, the sort of practical part of these studies that's really taking over in my field now is this idea that now that we get the bacteria to talk to each other, maybe we could actually impede the bad ones from doing that, as I showed you, but then also going back to the very beginning of my talk, remember there's all these wonderful bacteria that keep every other organism on this planet alive. Maybe the real way forward is to stop fighting against the bad ones and actually learn enough about the chit-chat of these good ones that we could make pro-quorum sensing molecules to get the good bacteria that keep us alive, or that are going to make our biofuels, or that make our medicines, or are going to make our plants grow in a hotter climate, make them grow reliably. Maybe we can learn enough about quorum sensing to make pro-quorum sensing molecules that make this planet and the people and the other living things on it healthier. And so besides the strategy I showed you, we are now looking for super autoinducers, right, to try to get begin this idea of not always thinking bacteria are bad, right, because that's what the bacteriologists have done traditionally as well. But now we're being enlightened that there's this whole other side to bacteria, and it totally needs studying. And then the final thing is to end with a confession, which is, now that we and lots of labs have this idea that we can manipulate quorum sensing, is to say, well, now wait a minute, we've been at this for a couple of decades. These bacteria have a 4 billion year head start. What if they must be out there doing this already? And so sure enough, if you go out and you dig up dirt, or you scrape stuff off a plant, the bacteria have already made all of these pro and anti-quorum sensing systems. So one guy will be out there talking with the molecule I showed you. The guy next to it in the dirt makes an enzyme that cuts the ring off the chain, and so the one guy's trying to talk, the guy next to it makes it mute. They eat each other's autoinducers, they make antagonists, they eavesdrop, they free ride, they do all of this stuff. And the idea that Bonnie Bassler and a couple of graduate students are going to make a better molecule is ridiculous. And so what we should be doing is just going out into nature and mining the bacteria for these strategies, because the ones that we find have won over billions of years of time. Those are the ones to bring into the lab and have the scientists start tinkering with. And so my lab has a big pony in that race, as do a lot of other labs, and it's really wonderful to just learn about these strategies that are out there in nature. And I think that's going to be a very big part of this field going forward. And then, of course, I want to finish by saying that I didn't do any of this. I have the good fortune to be invited by Jennifer to come give this talk. But this is my gang. And so somebody in that picture did every experiment and had every idea that I told you about. And what I would say, especially to the students that are here, the young people in the audience, if you notice, I'm the only one not that age. And what is so wonderful about the way science gets done in America is that it is always done by young people. So I always work with people that are between 20 and 30 years old, and it is so exhilarating and such a rush. They do not allow me to get tired or dusty or bored, right? And I think what's so great about this country is that whenever you read something that changes the way you think about something in science or engineering, right? It was always done by somebody who's young. And so any of the students here, if you think, science is interesting to you, I just encourage you to keep going, because our job is to think about things that no one's ever thought about before. It's the absolute horizon. I think of us as the adventurers of the 21st century, and it's really, really fun. It's loud. It's a little bit obnoxious with them, but I'm super grateful to that gang. And I'll just say one more time, find that I won't point them all out, but they made all the findings that I got to talk about tonight. And so thank you for listening, and thank you for inviting me and for giving me such a nice introduction. I'm just delighted to get to be here. And congratulations to you too.
So we have a microphone for questions. Should there be questions in the audience?
Hi, you were mentioning about the signal to the interspecies one, how it's conserved among all the species. I was curious about the receptors for that on different species. How well are those conserved? Is there evolutionary connections between them?
Yeah, so so that molecule, if you remember, it had a whole bunch of oxygens on it, and so the chemists in the audience, it can rearrange into different forms, and so but it happens on a very fast time scale. It's just rearranging all the time, and so the receptors are conserved, but they each recognize a different rearranged form. But if it drops out of the receptor, it converts into the other one, so I can talk to you and you can talk to me because there's a common intermediate, which is the molecule that I showed. Right? And so we can see how the receptors pluck out a particular version, and then we can see also the rules by which they talk to each other because of this fast interconversion. Yeah. But you can do exactly what you said. You can look in the genomes and you can find the receptors now because they look alike. Mm-hmm.
Scientist. But if I'm understanding what you're saying, you can go at something from both directions by making the good bacteria work harder and better, or the disease weaken.
Yeah, so that's the fantasy, right? Of course, right? We know we know a lot more about the pathogens, and again, that's historic. The scientists also didn't know all these good bacteria were on us and doing all these good jobs till literally about 15 years ago, right? And so much less is known about the beneficial bacteria than the harmful bacteria, because scientists have always studied these to try to cure diseases. But I think your idea would be, okay, so here's my fantasy is that you could have a cocktail, if we learned enough, you could have a cocktail of molecules, right? Where you're turning off the bad guy and you're enhancing the good. I mean, this is a long way away. But that idea, if we could learn enough about these, right, it's sort of like personal. Well, I'm an optimistic person, right? But yes, it is an optimistic. One of those people on the slide will do this, right? Anyway, but that is the goal in my field, is that so what I should also tell you is that my microbiome, these bacteria, are different than yours. You get them from your mom and dad, just like you get your human DNA, you get your bacteria, and we're all slightly different. And so if we could learn enough about them, you can imagine, this is what I imagine, these personalized medicine where I have this, cocktail of molecules for what ails you or doesn't ail you, right? And that's the dream in my field, right? Is that we could get these good guys to work even better for us. But we have to learn about them, right? But it's a really exciting time. It's like a renaissance in the bacterial field right now.
Great.
What about using these signals to make changing bacterial flora different? Because I know that having different bacterial flora will predispose people to different kinds of diseases.
Right, that's exactly right. So that's a more sophisticated line that I was talking about. So, right, so we all have these different bacteria. And so, you have to have the right bacteria in the right place in the right numbers. And then you're healthy, right? But all of these diseases that you're learning about, irritable bowel syndrome, come on, Bunny. Asthma, right? Crohn's disease. These all seem to have these whether you're obese or not, all have these microbial components, and this is just the beginning, right? And so the idea that you could use these molecules, right, to keep somebody down and let somebody else come up, that's there. And I just want to be clear, I'm here to talk about quorum sensing. Lots of other colleagues in the field are also trying to think about other strategies for how you make this community more healthy for people who have imbalances that cause these diseases, right? It's tricky because your human genome is playing with this bacterial genome, and it's a complexity problem on a massive scale, right? But others are starting to get those kinds of things to work in animals, right? in ruminants. Yeah, those are very expensive, right, to keep. So most of the studies are done in mice. And so, for example, there's a mouse that exists now that has, so you have these 10 to the 14 bacteria in your gut, right? 700 species in the right amount in a geography, right? These guys up here are different than those. It's always nice to have me after the dinner party. Anyway, so, right? Anyway, and so now there's a mouse that has 8 bacteria, 8 species, and it's really healthy, right? And so that's where I think right now the sort of center of mass of the field is. But you're right, we'd love to do that with ruminants because maybe you could make less CO2, right? You could make, right? There's all these dreams, right? But it typically starts in mice because that's what we keep in the lab. That's simpler. They're the mammal that doesn't cost as much to be, and you can have lots of them to try lots of different things. But it'll get there, right? It'll definitely get there, right? Yeah. Right, if they could get more, like if you can get more calories from the food, right? Absolutely, right? All of those are the dreams of this field. Yeah.
Hi. So you mentioned picking out this inhibitor based on whether or not it turned off the bioluminescence in harveyi, which is not a bad bacteria. So is that just a close enough bacteria to all the other bacteria? You can just use it for everything?
That is exactly what you got it exactly. That is exactly why we did it, because light is so easy to measure, and you can measure it very quantitatively over 5 orders of magnitude. And so it was just the first cut, right? Just ask who doesn't make light and take those, go from hundreds of thousands to a few thousand and then start doing chemistry and ask who crosses over into Pseudomonas. It was just the first cut. And so then now what we've done is we've made a Pseudomonas that makes light, right? And then we just do, right? We only do one thing in my lab, right? And so that's what we're going to try. We're going to go back and screen for better molecules, right? But we want to do it directly in Pseudomonas, and so we made a bioluminescent Pseudomonas that turns on light in response to its autoinducer. And I think that'll be actually a much more profitable screen to do, right? Because we don't have to, for the reasons that you brought up. Mm-hmm.
Hi, I heard you speak at Pebble Beach about 9 years ago at a Princeton conference.
Holy smokes, yeah. I was we've come a little ways. I remember that talk.
At that time, you used the phrase coherent whispering.
I did? Oh, I have to remember
which
that.
I thought
That's
was
good.
a lovely phrase, but now you seem to be talking about it in human language that is going more toward quorum, toward judgment on the part of the...
Yeah, right, because so I talk about it in human language because I think that's how I think about it. I think about what they're doing, but it is true. They are distinguishing self from other, and there's a logic, a decision-making, which is based on chemical reactions that allows them to do different things, right? And so we're actually thinking about it in a much more sophisticated way than a decade ago, right? So I think that they really are. It's not just 10 years ago we thought the molecule simply said number. Now what we get is they do that. They say how many, but there is information encoded in those molecules about species relatedness. Right? So there's an extra bit of information in those molecules, and so that's how we're thinking about it now. Coherent whispering. That was a fun meeting. Yeah.
Regarding the inhibitors for autoimmune 2, the universal one, if we were to find some kind of some kind of inhibitor and use it, won't it hinder the recognition between, suppose, E. coli and your own gut?
Yeah, that's great.
And inhibit your own enzymes?
Look at the time. No, yeah. yeah. No, you're absolutely right. So everybody get his question? Right? So you see how autoinducer 1 could be very species specific. If we could make this anti-autoinducer 2, it should be generic if we're right. And so you're right. So his question is, if you'll allow me to, right, is if we make that, you'll inhibit everybody, is it right? And so your point is very well taken, and that is exactly how it's supposed to work. And so what we're arguing, and this remains to be seen, right, anytime you take a traditional antibiotic, if you ever took penicillin, it doesn't just kill the good bacteria, right? What happens is that it kills like 95% of all your gut bacteria. So that's why you get a stomach ache, women get a yeast infection, right? And then the miracle of this microbiome is that you have what are called, right, it's amazing antibiotics ever worked now, right? So what happens is you have these persister cells, so you have these little pockets of cells, bacteria cells, that are not growing and they're not affected by the traditional antibiotics. And so after you finish the course of antibiotics, they repopulate and your microbiome comes back. So that's been going on for 80 years, as long as we've been taking antibiotics. And we are banking on that in the autoinducer 2 strategy. So it's so much worse, where you kill these bacteria. So you're absolutely right. If we can make this fantasy anti-autoinducer 2 molecules, everybody will be goofy, right? The pathogens will not release their virulence factors. The immune system's job is to know good from bad. It hunts them down. It gets rid of them. And then everybody goes back to talking. We have to prove that. But we're using what's known about traditional antibiotics to be pretty confident that, in fact, you don't have to be that selective, right? Because we never were, right? And that's sort of what's amazing now that we know about it. And so that's the strategy, right? But again, we're at the same place with the anti-AI2 molecule as the anti-AI1 molecule, right? And so we need to really prove that the bad guys get cleared and that the good guys come back, right? And the molecules have to be a lot more potent before we can do such an experiment. But we're getting there. Yeah, but thanks, that's a great question.
Rather than building the resistors that would jam the bacteria, wouldn't it be easier, or would it even be possible to build a bacteria that would eat another type of resistor?
Right, so this goes back to these questions. If you could sop up either a bacterium or make an antibody, right? If you could slurp out the molecule, right? That would be fine, right? So we did the screen, and what we got is what we got, right? We got a molecule that interacts with the receptor, right? But absolutely, if you could get a molecule that shuts off the enzyme so they never make the autoinducer, or if you could make something that either a bacterium that eats it, but it's got to eat it all, right? Like really efficiently, or a molecule that takes it out, like makes it pellet, come out of solution. Those are all fantastic strategies, right? And so now that we saw that this one is starting to work, we can actually design screens to get the other kinds of molecules. And I don't know which, if any, is the best strategy in therapy, right? But we're trying. But you're absolutely right. That is, people are hunting. We included for that. And then I should also say, I forgot to say about like in industry. It's like now you can imagine like putting these anti-quorum sensing molecules like in the materials that make catheters and intubation devices. You know how you get an infection, you get a biofilm, right, on your stent, on your knee, right? So there's beyond just a medicine. You could put these in paints, you can put these in contact lenses, like all these things where you either want bacteria to come or you don't want them to come, right? And I think the industrial, I don't know why that your question, remember, the industrial things are going to happen faster because you don't have to put it, you don't have to eat anything, right? But I think that all of these kinds of strategies, either they don't make it, somebody takes the molecule away, right? Or they can't respond to it. In all kinds of tactical situations, one of those strategies is going to be best, right?
All right, well, let's thank our
Thank you
speakers.
for letting me come here. It's really fun.